Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts. And now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 323. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone, and trick or treat.
Trick or treat. Welcome to Halloween. Also joining us from Ann Arbor, Michigan, Michelle Swanson.
Hello, good to see you all.
And from St. Louis, Missouri, Petra Levin. Welcome.
It's great to be here.
Our guest from the University of Puget Sound in Tacoma, Washington, Mark Martin. He was mouthing the words for me because he knows I always get it wrong. Welcome back, Mark.
It's always good to be here, and happy Halloween to folks. I wish you could see all the things in my office. I'm waiting for Petra to make fun of my giant microbe with the skull mask.
Why would I make fun of it?
Because you did last time because of the fez.
Oh. The
I
fez.
made fun of the fez.
I did not make fun of the fez.
You
do not put it on me. It's Michael.
I think that's too small, Petra. It's great to be here.
TWiM adjacent Mark, right?
That's correct.
Doc Martin. And before we get into the science, which is cool today as usual, maybe even cooler if it's possible, I'd like to remind you, if you enjoy these science programs, we'd love to have your support to make them, because we do not do ads. We only exist on listener donations, mostly individuals like you.
And nor do we collect a salary.
Don't. And we have a few corporate sponsors, but not very many. The rest are grassroots. So November and December of this year is our big fundraising drive. So please be generous. And if you like, science programs, if we appeal to your inner science self. And by the way, if you like these programs so much, send us a little audio telling us why you love them, how it's changed your life, or how it appeals to your inner science self and so forth. And you can send it to incubator@microbe.tv. Otherwise, if you'd like to make a contribution during these months, microbe.tv/contribute. Okay. Oh boy, our first offering is about concrete, and it doesn't involve the mafia. Michelle.
Yes, and it is titled Construction Nanobiotechnology Approach for Performance Enhancement of Microbial-Induced Biomineralization Using a Biopolymer Encapsulated Spore-Based System. And this is the work of Ankita Debnath and Ramkrishna Sen. They're from the Department of Bioscience and Biotechnology at the Indian Institute of Technology in Karapagar, India, and it was published in Applied and Environmental Microbiology in September of 2024. So concrete. I will start with a personal say that my dad was a chemical engineer, first in his family to go to college, and then he was in the cement industry for his whole career. So I learned at an early age that concrete and cement are not the same. They're not synonyms. Concrete is a component of cement, which of course we use across the world for many functions. Roads, of course, dams, bridges, foundations of buildings. So it is very malleable. You can make it in different shapes, etc. And Their estimate is more than 4 billion tons of cement are produced globally each year. That's the good news. The trouble is that production of concrete is responsible for what is an estimated 8% of our global carbon dioxide emissions. So there's definitely motivation to make sure that the concrete that's made is long lasting. But as you probably have experienced, concrete can crack, you can have fractures in it. And so for more than 20 years, scientists have been working on self healing concrete. And that's what we're going to focus on today. So, when concrete cracks due to temperature changes, just heavy loads, humidity, etc., these small cracks can open up. Water can enter, and then if is a mechanism to generate calcium carbonate, those little crystals can fill the cracks. So the strategy here is to identify microbes that can survive the process of being mixed into the concrete, and then when water flows in, trigger their production of minerals, including calcium carbonate. So that is the magic that we're going to describe today. And the microbial hero is a bacterial species called Panabacillus alcaliteraria, I don't know, something like that. And it has the benefit that it naturally forms spores, which of course are very hardy cell type, and also that it is a producer of biominerals, including calcium carbonate. And its favored growth pH is up around pH 9.5, which is ideal because the pH of concrete is also quite alkaline. So these investigators, in part, used their own research, but also built on the literature to examine whether they could optimize use of this Panabacillus in self-healing concrete production. So they first wanted to solve a problem, actually, because even though this bacterium naturally forms spores, the shear forces that are created during the process of mixing concrete, and many of you have no doubt seen those big trucks with the compartment on the back that's going round and round mixing the concrete. But during the production of concrete, the shear forces and the heat can be quite extreme and damage the spores. So then they're not available to work their magic. So what these authors have done is focused on coating these bacterial spores with a protective extracellular polymeric substance. So this in our field is abbreviated EPS, and these are really very common in the living world. Microbiologists will think perhaps of biofilms, which are colonies and communities of bacteria that practice division of labor, but they are protected by a coat of polymer, often called an EPS. And that's just one example. So what the authors have chosen is a EPS made by a bacterium, Cellomonas flaginea, which they are able to culture using material left behind in the sugarcane food industry. So when sugarcane is processed to extract the juices, it leaves behind this pulpy material, which can be used then as a food source for the bacteria that are going to make the crystals to cure the concrete. So that's an advantage because that too is a green step. Like we'll be able to use what is normally a waste product to culture the bacteria and therefore reduce the cost of this self-healing concrete. So the first thing they wanted to do was characterize the interaction between these spores and the EPS from the sugarcane culture. And to do this, they used a lot of microscopy, fancy microscopy, but also a lot of spectroscopy. And I won't go into the details because I don't have that expertise, and it's also a snippet. But I will tell you they started by just in one of the figures, they show us what the structure of the spores looks like, what the structure of the germinated bacteria look like, and then the EPS. And so in figure 5E, we can see that there's a yellow arrow pointing to the spore, a red arrow points to a spore that has germinated to form a vegetative or replicating bacillus, and then a blue arrow is pointing to calcite biominerals, which again is the magic. That is the mineral that can fill the gaps in the concrete to have it self-heal. So many of the tests that they used first wanted to determine whether what was the pH that would be optimal for germination of these coated spores. So in figure 5A, for example, they compare pH 4, pH 7, pH 9, and measure germination and then also growth. And fortunately, what they find is the optimal pH is up in the pH 9 to 9.5 range, which is great because, again, the concrete mixture is quite alkaline, but also that is known to be the temperature where the extracellular polymer substance will solubilize, release the spores, which can then germinate and then begin to grow. So far, so good. They then are interested to see whether the bacteria within the spores and coated by this extracellular polymeric substance can withstand stress. Are they really more resilient? So to do that, they made a suspension of the coated spores, and then they subjected them to heavy shear forces created by vortexing for 5 minutes or as long as 30 minutes. And then they tested their viability and also looked at their structure. They also allowed these coated spores to sit for 3 days and then looked to see whether the spores were coming away, being released from the EPS. And fortunately, they found even with the heavy vortexing, heavy shear forces, they saw less than 1% leakage of spores from the coated mixture. So that's very encouraging because, as I said, that's been a major challenge in this biohealing concrete. They then looked at the impact. Oh, I already mentioned they looked at the impact of pH on spores, and that was all looking good. And then they wanted to ask whether the EPS coated spores can actually produce the calcium carbonate, the small crystals, which would then act as the structural component that would fill the gap. So they cultured the coated spores in media that mimics concrete. And we can see in figure 5E that I mentioned earlier, we actually see that the spores can germinate and then form the calcite biominerals. They also measured directly, and again, using really sophisticated analytical chemistry techniques, what was the rate at which free spores can biomineralize and form the calcite, and also compare that directly to EPS-coated spores. And fortunately, they found that the EPS-coated spores could biomineralize at a rate almost double that of the free spores in their in vitro setting. And they imagine that that's even an underestimate because they are not able to subject their spores, coated or not, in the laboratory to the same stresses that they'll experience during the process of making concrete. They also used electron microscopy and also, again, some analytical chemistry techniques to confirm that indeed calcite is being formed. And if you look at figure 7, figure D and E, you can see electron micrographs that show the structure of these calcite crystals. So in general, this was a very systematic study where they first characterized production of EPS, looked at what kind of side groups were available. They then did some more analytical chemistry and deduced that there were lots of interactions that would stabilize the binding of the EPS to the spores, mostly through ionic interactions, because the spores are very positively charged, the EPS is negatively charged, so that accounts for the stability that they observed during the heavy 30 minutes of vortexing. They then verified that the bacteria could still germinate and begin to grow and produce these calcite crystals under conditions that mimicked the best they could the conditions in the concrete industry. And they used lots of analytical quantitative chemistry to look at the morphology of the interaction, look at what chemical functional groups were there, whether they were in crystal phase, liquid phase, whatever, what particle size there was, what was the composition of the elements in this final product. And so they are left feeling quite encouraged that they've made some good progress in finding a way to make these spores more resilient by coating them artificially with the EPS that they have harvested from another microbe, and found that they are indeed quite resilient, and that this is another step forward to advance this field of biomineralization and making self-healing concrete.
Michelle, in the review you had sent, they also mentioned that making concrete has a high carbon footprint.
Yes.
So much of it is used. It's like 6% of the carbon on Earth is from concrete. So maybe this can help with that as well.
You can repair instead of make new.
Yeah.
I mean, there's also all these buildings made out of concrete that are kind of famous from the 1960s and 70s. They're dissolving. But actually, what I thought about this, because I recently went to the dentist, and they have to scrape off a lot of this calculus, a lot of biofilms, bacillus biofilms, for example, which are not forming in your teeth, but apparently strep mutans. They also make this concrete-like substance that they scrape off your teeth. So it seems nice that we could use it to actually not just be gotten rid of, but to help resolve. I thought that was really cool.
Yeah. That would be called calculus.
Exactly. And it's calcium phosphate mostly.
Yeah, it's a nasty little creature. And Michelle, when you were growing up, did your father ever use the term clinker?
Yes. Oh, yeah. That went into the kiln.
Yes. and so here we have microbes interacting with clinkers, making their very own clinker without the carbon footprint, because clinkers are the essential magic component of Portland cement. They come together when they mix the two. They mix the limestone from that region of England, and then they mix the other silica with it, and then they heat it up to a really high temperature to make these clinkers. And then when you add water, you get the concrete. And so what we're having is the bacteria are actually making clinkers. Who knew? Who knew?
Yeah, it's really a wonderful interdisciplinary study of applying microbiology to chemistry and then engineering to try to advance not only our ability to build in a more green way, but also to reduce our carbon footprint.
And Michelle, as I was going through looking at other things, I found this other article, Microbial-Inspired Self-Healing of Concrete Cracks by Sodium Silicate Coated Recycled Concrete Serves as a Bacterial Carrier. And this was in the Frontiers of Structural and Civil Engineering. So this is a big field that's emerging, and it's not something that I would have ever brought up in any of the micro classes that I'm introducing students to. And I'd be interested to hear whether Petra or Mark introduce these topics to their students in the undergraduate years.
Yeah, I've talked a little bit about that. Students are always interested in the structural aspects of it. I did want to ask a question though of Michelle. Have you heard of Roman concrete?
I haven't.
So I actually have a link that I'll share on the show notes if I'm supposed to do that, about Roman, which is also self-healing, and I think that it's based on using volcanic ash to make it possible, because those roads have lasted for a very long time.
Yeah, and I think it's the water seeping into the cracks then triggers the chemistry. Is my understanding.
And wouldn't it be interesting to learn that once again, microbes are just doing what nature does in other ways?
Or maybe microbes are doing it for nature. That's the part we don't know. Yeah.
First evolved, last extinct.
Exactly. I mean, I do tell my undergrads that microbes produce like the calculus on your teeth. But Yeah, this is,
But
I mean, I like, that's not, applied.
But the idea of microbes on building materials, there was, I think it's Noah Fear who did this. I've forgotten the associate's name, but Noah Fear was the PI about the microbiota that you find on gravestones relevant to today. And you find granite versus concrete, you get very different things. And they actually follow geographical differences as well. Where, like, if you go to Germany, what you find on a granite one is similar to what you find in Cambridge, Massachusetts on a granite one. And it's very interesting to think about that role. And that's why I tell students that the idea of geomicrobiology is so important for that reason.
Yeah, I mean, I learned about it also, I think, from Diane Newman in a seminar she gave like way back about rust on iron and that kind of building material. So very different building material, but that the microbes really facilitated the oxidation steps. Microbes, once again, important.
Why microbes are amazing, right?
Yeah.
Well, and I have to credit the scientists who are thinking about exploiting or leveraging these bacterial processes.
Yeah, I thought this was a great idea.
Yes, we see no lack of creativity in these applications. It's really great. Okay, now we go from concrete to bioluminescence with Doc Martin.
Oh, I love this topic so much. I think that this particular episode ought to be titled Light in the Underground, because that's relevant to what's taking place in this paper. Now, I do want to give because this will make Michelle happy, the title of the paper we're discussing is Bacterial Bioluminescence is an Important Regulator of Multitrophic Interactions in the Soil. And this is in Cell Reports, and it was published on October 22nd of this year. The authors are Mueller, Morales Montero, Boss, I wish I had that name, Hiltman, Castaneda Alvarez, Bott, Arche, Glauser, Joyce, Clark, and Machado. And they are in, and I hope I'm pronouncing this correctly, in Neuchâtel, Switzerland, and also Cork, Ireland. And this is an absolutely dense and interesting paper that will raise so many questions. I am delighted to talk about it with you. I also like how the authors would say, we don't know why this happens, which I enjoy seeing. The first thing that I want to remind the listeners is that this is an example of a journal that gives a visual abstract. And what I like so much, it's on the first page, is I love the descriptions of positive and negative interactions. And they give highlights, they give it in brief. So it's a wonderful way to start the dive in. But I want to warn you, there are 169 references in this paper. That's a lot.
We should also say this paper is a team effort by 3 first authors. It's dense because in part it is this big team effort from Mueller, Morales, Montero, and Boss. And I think that's really kind of cool because not all the time do you read a paper that has sort of these 3 people working together. And I think that's what makes it so strong and so data intensive. So.
Yes, and different approaches as well. I think we tend to silo ourselves. For example, plant biologists look at questions differently than microbiologists look at differences differently than, say, a behavioral ecologist, all of which is factored in here. But the first thing that I want to say is that I owe a lot of my interest in this topic to the great Heidi Goodrich Blair, with whom I've known for many, many years, and I have samples of this particular organism. I've grown the nematodes. I have many thoughts on this particular topic. So to give a quick thumbnail sketch, one of the biggest mysteries in bioluminescence is a simple one. When you talk about bacterial bioluminescence, you find it very commonly in marine environments. I've never heard one in a freshwater environment. I have heard about it in estuaries, and one example in terrestrial. Now, there could be many reasons for that. I'm a big believer in cryptic bioluminescence. I'll come back to that. It could be people just aren't looking. And that may be the case. What's interesting here is Photorhabdus is the one example of a terrestrial bioluminescent bacterium. And for listeners and viewers, I put some information in the show notes that is really interesting. Photorhabdus has an interesting association with nematode worms. To be brief, what happens is that they are carried by the nematode worms. The nematode invades insect larvae. The bacteria are released. They create a number of antimicrobial agents and other types of compounds that are able to affect the insect host. And then what happens is they procreate in there, and then eventually it's just a bag full of nematodes, and then they're released. Now, again, the bacteria have formed an association in the pharynx of the nematodes to do this. Now, I've had students bait to look for nematodes quite often. And you use like waxworms, which is what they use in this study a lot. It's a really interesting topic, not necessarily germane to this, but I wanted the listeners to understand about this life cycle. Again, the one example of a terrestrial bioluminescent organism, and that's important. One, there may be others, and they talk about the fact that this particular genus is really common worldwide except for Antarctica. And it's important to think about that.
I should also mention that I teach this, and I think you might teach it, because it's both a pathogen, right? And it's a mutualist, right? Because it has this mutualistic relationship with the nematode, but they can team up to infect these larvae. So that what Heidi Goodrich Blair used it for, and other people, is to be able to decide, like, what's the difference between being a beneficial microbe and that same microbe being not so beneficial.
So as I've said to Arturo Casadevall more than once, it's always context.
Exactly. But you can see like which genes are important, right?
Kind of like location to a real estate agent. So what I sent all of you was a GIF that I made of some of my own work with that, where I was micro-injecting waxworms on the one side versus saline in the other, and they do indeed glow in the dark.
Yeah.
And it's what I've always used it for. And students love it, because who doesn't? By the way, you want to do this with waxworms, not with tobacco hornworms. It gets really gross and floppy. I just got to tell you. I know.
You've tried.
Oh, yes. Are you kidding? It's like you don't know me. Okay. Now,
no, we know you.
So what I want to say here is that I was really surprised, because as I started to say a few moments ago, marine bacterial bioluminescence is extremely common, but in terrestrial cases, not so much. And in fact, there's all this mythology. I put a link up to that. And because it's Halloween, do you all know about angel glow from the Civil War? There was a story that's been going around that during the American Civil War, people had noticed, because there weren't artificial lights, that people with terrible wounds, the wounds sometimes would glow faintly in the dark, and then those people would get better, hence the term angel's glow. Turns out that's most likely Photorhabdus making these natural antibiotics, except as I dug into it, those killjoys at Snopes suggest that's not the case. It is true that Photorhabdus makes a wide variety of antimicrobial compounds. And as we were just discussing a moment ago with Petra, it's certainly involved with the mutualism that it experiences in the nematode.
So let me ask you a question, Mark. It goes back to the hypothesis why you see light-producing microbes mostly in the oceans as opposed to terrestrial environments. Could it simply be that the microbes have a much better alternative electron dump in terrestrial environments than they do in the oceans, where their anaerobiosis is much more extreme and they will not have been selected out to go into a different area of the water column where there may be oxygen, which will give them an electron acceptor. And light production is a way of getting rid of excess electrons, especially when these organisms are using the excess carbon that may be found in the water column, whereas in terrestrial environments, carbon may be more sequestered, and so there may not be a need to dump the waste electrons into something like light.
Well, you might be right. All I can tell you is that I've been wrong with the point of view of microbes for many, many years. They have their own agenda, and it's not mine. So all I can tell you about it, it's been a mystery to me for many years. I would suspect there are many other bioluminescent terrestrial bacteria that we haven't seen yet.
But what could they be doing, Mark? What could they be doing?
This comes back to the most important question: why make light? Now, you heard Michael say that it has to do with dumping off extra reductant, and there's some evidence for that. In the body of the paper, the authors talk about some things that, and I want to be very gentle when I say that, have not been borne out by experimental data in this area. For example, some have hypothesized that light production could actually power photolyase to repair dimer-dimer formation. And that was work done by Eric Stab, who showed, no, that's not what's going on. But it is certainly worth thinking about. And I'm delighted that they have created all these references for me to smile and remember all these great papers on. So let's get to what they did. I believe, to answer Michelle's question, I believe that bioluminescence has a biochemical function and light is a byproduct. And I think that when the light is happening, then that's something natural selection can act upon. And I have many reasons for saying that. So what I like about the paper, they do a nice job in the introduction. A figure 1 that they put in here is a nice, very confusing circular diagram, which I know I can sense their excitement when they put it together. And I think if you take your time looking at it, you can actually see where they're going with it. It reminds me of what they tried to do in the visual abstract they put together. So the first thing that I want to deal with in their results is they're finding that the bioluminescence production is, they call it an evolutionary dynamic process. Absolutely. No matter where you look for bioluminescent bacteria, you find some that make a little light, some that make a lot. There are some that are cryptic. All of these things are true. What's interesting here is they got 59 strains of Photorhabdus from all over the world. They went ahead and, as you can see, they assembled all this wonderful work you see in figure 2 showing relatedness between them. And I didn't know there were that many isolates, which is kind of cool. And they have them now, which is wonderful. And as you can see, when you go through that, they're showing that different isolates have different levels of bioluminescence. Something they addressed a little bit obliquely is that Photorhabdus is an unusual organism. It throws off variants quite commonly in terms of colonial morphology, and some of those variants aren't as good at colonizing nematodes than others. So I think it's a very dynamic situation. The reason that they're showing you this is they're showing you, and by the way, the whole idea of putting infected larvae in CoStar dishes and using a visualization to, you actually see this, I guess, in part C, different isolates, different levels of these poor infected larvae are just wonderful. So in any event, they're showing that they have a wide variety of luminescence that's produced. And by the way, there are plenty of examples of bacteria that do not produce light at all that form symbioses with nematodes. So this isn't absolutely there. By the way, because Michael likes to throw questions at me, do you have any idea, Michael, why bacterial bioluminescence is at 490 nanometers?
It's a safe wavelength.
It is also the wavelength that transmits best through water.
Okay.
Yeah,
but then that
blue-green
would suggest this is not just a coincidence. This is important.
and you know, it's interesting because, again, they came at it from far away from microbiology, I think. They had some members of the team that are microbiologists. And all the bacteria that I know that produce light, and I'm familiar with all of them, they're pretty much blue-green in terms of the light they produce, about 490. Now, there was one example that Ned Ruby had of one that was yellow, but that was one that had a photochrome that absorbed the light and then retransmitted it at a different wavelength. So, and they do say that perhaps this moved to the land from the ocean, which is certainly possible. But if you look at figure 2, when they show so much differential, I can promise you that when I pick, as I often do, Photobacterium phosphorium from seawater samples, I get wide variation in the amount of light that gets produced. There is a difference here. They don't say this, and I will, that when you first isolate bioluminescent bacterium, it often is unstable. And this goes all the way back to Biernik in Delft. And so what people tend to do is stick with something that seems stable. And I believe all 59 of their strains were ones that have been well characterized. And that's good. By the way, that instability takes back to Michelle's question, what is the functionality to this?
Yeah, and if I could just add another point to that, they do make the point that bioluminescence has independently evolved more than 90 times.
Yeah.
So it's not just our favorite microbes, but yeah.
No, I in fact, I have a petunia plant that is bioluminescent now. That has fungal bioluminescence genes in it, which is just awesome beyond words. Mm-hmm. You know, if it glows, I'm in. That's what it comes down to. You know, so it's interesting because Photorhabdus has the typical setup of genes, and we'll talk about that in a second. I wasn't able to find out if it does quorum sensing. Now, I didn't go look through the genomes to look for quorum sensing genes. I believe they have what are called Luxar solos. That is to say, they have some elements of it and not others. But I know a lot of people like using it when they make gene constructs because it doesn't undergo quorum sensing. So in any event, so they showed all of the, see, I'm doing the they thing. The researchers were able to show all this variation from their different isolates, which is great to know. And then and this is the one that really got me, really grabbed me by the throat and shook me like a dog with a snake. Sorry, Carl. This is an element that I'm concerned with. And the idea is that bioluminescence, I'm glad they couched this carefully, may alleviate oxidative stress in Photorhabdus. Now, since I work with bioluminescent bacteria in my copious free time, I can test that with some of my own stuff because I haven't done this. That is to say, they're showing that if you're making light, you are more resistant to hydrogen peroxide. And they really beat that to death, you know, with a flaming tennis racket. They do such a nice job of showing that the data is real that I trust them with it. They also were able to create individual deletions of individual genes. For example, the Lux A and Lux B genes create the actual subunits of the luciferase. The other genes in the operon generally involved with the fatty acid reductase and recycling. So in any event, this is really interesting because many people have talked about a biochemical function for bioluminescence, not, it signals, and because that isn't how it happened. This is interesting to me. That's why Eric Stabb's prior work, trying to see if this is important, might be part of it. But they did a really nice job. They also, and I had never heard about this, and if you all have, please be gentle with me, the idea that light can directly break down hydrogen peroxide. Yeah, the bottles are dark.
Oh, yeah, the bottles are dark. You're supposed to keep it in a dark place. That's exactly right.
So what I love about these folks, and I don't mean that in any negative way, is they say, what kind of controls can I do? And for many of their experiments, they can use like a knockout in, say, a Lux A and Lux B gene, but they'll also have little granules that they charge up with sunlight that are completely abiotic as a source of light. Long story short, what's interesting here is it looks as if the light itself being produced can break down some hydrogen peroxide.
Yeah, that's very convincing.
Yeah. So and I again, this is a very dense paper. There's a lot of information, and it has, and I'm in no way critical, a lot of supplementary information. So if you aren't used to looking at supplementary information, I really recommend it with this paper because there's so much there. So
Mark, along those lines, when microbes are challenged metabolically, internally, they often go into oxidative stress. And so the excess electrons that are going into the membrane, if they can't get out of the bacterium, the electron will effectively add itself to an oxygen molecule or a water molecule, making free radicals. And so this is perfect because the light that they're producing is going to inactivate the hydrogen peroxide that the metabolic stress of not being able to get rid of the electron is going to accomplish. So I think you and I are along the same lines of answering why make light.
Well, In another context, I'll tell you, I worked specifically on this question with another species of bioluminescent organism, and this has given me so many good ideas to follow up on. I do want to say parenthetically that, and I don't know why they did this experiment, but I'm glad they did. They wanted to take some of their strains and show that when they did in laboratory evolution, they would get changes in the amount of light that's being produced and then find out what genes were involved with that. And they have a lovely chart of all that. But what was interesting is that when I do those experiments, things that aren't spending energy on light production grow faster. So when I do those experiments, I get things that don't make light as well. But they didn't do that. Now, it could be that they were doing 1 to 10 dilutions and I do 1 to 1,000 dilutions to give things a chance to grow. Who knows? The point is they did exactly the kind of stuff that I try to do, and they get different results with Photorhabdus. And it could be that Photorhabdus is just a different beast. And it could be what we're going to be finding out is taking place later in what its actual role is in the underground. And that's something I had not anticipated. I would have never guessed, because I knew how few Photorhabdus are. I mean, you can isolate them, but I didn't think they were a major component of soil microbiota. But they have quite an effect. And so what you discover is that the roots are able to perceive light being produced by Photorhabdus. Now, when I say that, generally speaking, I'm talking not so much about the bacteria, but the infected nematodes. And there are some remarkable images to share. So what you find out if you look at figure 5 is plants are responding to that Photorhabdus bioluminescence, and in turn, that impacts their growth, and they're resistant to root, but not leaf herbivores. Now, I saw Petra's face, and that's how I felt. Petra, this was me with the whole paper.
I mean, it's amazing because it's one of those things where you're doing the experiment and then you get the result, and the result is super informative, but not at all what you
expect.
Yeah.
Yeah. That's what Bill Metcalf told me a long time ago about phenotypic tests and you doing bacterial genetics, is that, your phenotypic test, it doesn't matter. The bacteria have their own agenda.
No, and I mean, we talk about that. I'm in a department that has a lot of plant biologists in it, and we talk about a lot of herbivores. There's ecology, and then also straight up plant biology, and how that responds during the day and what the signals are. But this is actually I've never thought about luminescence as something in the rhizosphere would modulate the plant from the roots up. You kind of forget about the roots when you think about herbivores, right? You think about actually, first of all, animals, right? The deer aren't doing anything, but even the insects, you really don't think about the roots at all.
And for me, Petra, I realized I haven't turned on the luminometer and just gotten some soil and put some soil in there to look.
Exactly. Of course, it's dark.
I haven't looked.
Well, but soil is not sterile, right? There are lots of critters,
microbes
and insects, and
Yeah.
And fungi, the whole mycorrhizae down there. But I don't know how many of them are actually producing light. But this is...
I don't know either.
This is sort of giving vibes of a plant version of cordyceps. It's taking over.
Yes. Yes. And then you start thinking about optogenetics on top of it, right?
No,
I know. So the important take-home lesson is those roots are watching. Those roots are watching. And when they see the light, it's using the available mechanisms that plants use in their deals with potential herbivores, is the take-home message that I got. And they, again, took that flaming tennis racket and they just beat this to death using so many different techniques to find out what's changed and what hasn't. And to me, I can sense how amazed they were with some of this. Now, when they talk about Photorhabdus impacting insect growth, I expected that, but not resistance to nematode attacks. You'd think that they would try and keep other ones from getting in, but they're not doing that. But it's via using the plant. So it's more of a partnership. And this was fascinating to me, but not as fascinating as what I'm going to get to. So who has some questions before I go to what the really cool stuff is to me? Did you hear me say to me? Yeah.
One question I had about plants, and this is maybe not a question for any of us, but in the roots, right? how are there must be photoreceptors in the roots. Yeah. It's just I always think of them again in the leaves because they're the ones that
I absolutely, they have blue light receptors, and they have another receptor I can't think of the name of, and it always made me wonder about people who were doing hydroponics if they thought that through, right?
Oh, yeah.
But it kind of makes sense in a way. And Petra, it sounds like you're thinking the way I am about that. I hope you don't mind my saying so. How much light gets through the soil? I'm not thinking about that. Right.
Well, I mean, a lot of people who work in plants, right? You talk about shade, and then you have like light receptors, and then you have red light. The shift that happens in the shade from the algae. This is a big issue in green algae, the shift in light and things like that. So I don't know like what the roots are seeing, but now I'm now going to go bug everybody in my department who works on plants about that.
So I want to emphasize again what it seems to me. And nor am I a plant biologist. I once did some work with Arabidopsis, but that doesn't really count. That's E. coli with roots. I understand that. But, kind of is.
But it's a plant. It's
a great plant.
E. coli is a bacterium. It's just not all bacteria.
That's right. I got that from Esther Anger. Did you hear about this, Petra, where she said that all bacteria are not like E. coli. In fact, E. coli isn't like E. coli. And I love that.
No, that's true. But it also goes back to Socrates, right?
Yes. So
all bacteria are microbes, E. coli.
What I found really interesting is, let me see what page this is, because I really like what they work through. So glad this is open access. On page 7, Photorhabdus bioluminescence acts as an aposematic signal against scavenger attack. This blew me away. And the reason why is if you do fishing in the ocean, and you have something that's glowy that you throw in, the fish hit it. So much so that people like me think that that's how marine bioluminescent bacteria go from fish to fish because of the fecal pellets glowing, because they're colonized by them. But this is the reverse. It actually makes the scavengers run away from the glowing infected in carcasses. I guess the carcass is the right word. And I love the fact that they tried a couple of different predator bacteria, that's so smart, predator insects. And it really seems to act as a protectant that allows the carcass to finish its development to release the nematodes. In case you didn't know this, and this is for you, Michael, when the cycle is done, you have this brownish dried up carcass and you put it on a damp piece of filter paper and all the nematodes emerge while you watch.
Ooh.
And when I used to
good thing this is Halloween.
Big. Yeah.
They're
and
like
what
tiny.
I loved, I had a student, I used to have students do this, and I had this one person who wrapped the plate she was doing this in about 15 layers of parafilm because she thought they were, and I wanted to say they're already on you, but I didn't say that. But the fact is, that is how you protect the site that allows them to actually do their business. You'll notice that they even used, they're showing that it's the light nearby that matters. They put some eggs next to the carcasses, and still the predators left them alone. And I thought that was really amazing.
Yeah. I wonder if that's just serves as like a microbial associated pattern. And so they're like, I'm staying away.
A PAMP, yeah.
Yep.
Well, it's interesting because, again, to me, I'm used to the marine system where predators are attracted to light and consume it. And this is like, no, I don't want to go anywhere near that. And I think it has to do with the life history of what we're dealing with, where you have a colonized carcass that's developing all this stuff. This is a paper, by the way, that I really want to recommend to students of mine to read because there's so much in it. I could say I wish they wouldn't say aluminescent for something that's dark, but I understand why they do that. And they actually have some evidence now that when they used light deficient Photorhabdus, that there's some changes that take place with the symbiosis with the nematodes. And this is a nice counterpoint to what I know takes place with Euprymna scalopes and Alivibrio fisheri. I'm supposed to call it that now. And how if they're dark, you don't get quite the proper symbiosis. So there's that interplay between things that I so enjoy. And I think it's similar in the sense that when you have a light organ being colonized, that's similar in essence to having a carcass being colonized.
It is colonization for sure.
Yes.
and
so there is so much there, and I'm getting too enthusiastic again.
No, but I think this is actually really interesting because the whole light, and you will, Mark, know a lot more than me, the light and the squid, it's they always say it's to hide them from the moon, and I don't know if there's actually any data or that's just yeah. Okay. Yeah, we're waving our hands here. You can't see us, but that's what
I'm
we're
sure
doing.
Margaret would disagree. But
is there experiments, or is it? I mean, this is a why question in biology that's always very difficult. Now, obviously, the squid system is more difficult than a plant system because you can't just...
The squid move.
Yes, and as we know from every plant lecture I've ever attended, plants don't move. But yeah, so that's I like that also here, that they kind of get in. They start to get towards the answer to why in a experimental way.
So the idea of like projecting light downward to appear like starlight to avoid predators is something that I have no problem with, and Margaret has very strong ideas about it, and I generally go with Margaret on things, which is the wise move.
Oh yeah,
but no, because she knows a lot.
No, she knows a lot, and this is partly you just have to go with how you understand the system. And she
so when you're
talking
talking
better
about
than anybody.
Right. And there's absolute knowledge that there are light receptors in the squid responding to the light being produced. So in essence, this is a similar kind of approach. But with plant roots, I hear my voice is too high, but you know what I mean. I say this all the time. You probably heard, I'll OMG for overwhelming microbial greatness, and WTM for what the microbe. That's where I'm at with what the microbe here, because this is just amazing stuff. And what I really like is the author spent some time saying what they didn't know.
Yes,
so
also great.
What I could see is someone seeing, I'm used to what I used to call the minimal publishable unit, where it's just like a tiny paper with a tiny set of things, and there's so much here. But clearly this group of people interacting, I wonder if they all have flashlights and they're all signaling each other, but that's me. So where I'm going with this is they clearly are really excited by this work. I think it comes through in the way that the paper is written. I think it's a wonderful source material for how to do different kinds of experiments. So if I wanted to know how to go about testing for avoidance, say with the bacteria I work on, now I know how to do that. So there's not just really cool experiments, but a good guidebook. What I particularly like is they forged their own way in many ways, at the same time giving careful credit to people who've done a lot of the work.
Can I also add that they conclude at the end of their introduction that our study thereby sheds light on the evolution and ecological relevance of bioluminescence.
No, I really like that. And I want to add that in the communication from the senior author, they emphasize this teamwork that we talked about.
Yeah.
But actually, now that, we've gone through the paper, you can also think about teamwork from an intellectual standpoint, where you're really getting ideas bounced off each other. And that would never have, that's harder when it's just your PI and one person. But this idea that you have a team, and the PI's advice for new PIs was that teamwork is a big focus in his lab. And that's kind of how, working together to solve the problem. But also that he recommends, he started his lab during COVID, but he recommends that the literature, which clearly they know the literature across like several different organisms, which is impressive. And don't be concerned about time. Sometimes answering hard questions is difficult, but also because of that, don't shy away from the hard questions, because this paper really, gets me thinking about this bioluminescence in a way that it's not just incidental or has a potentially, a mechanism. They actually have not a mechanism, a reason for being, but here it's sort of they can test these things, and that is super impressive. And I'm excited to see what comes next.
And did he also add that this was 5 years?
Yeah, it was 5 years.
An endeavor
right?
that took them 5 years. Yeah.
And that's why he emphasizes, like, it's more important to take, and this doesn't always work with funding agencies. Maybe the Swiss funding agency is a little more resilient, but patient. But the idea that, you want to ask big, important questions because, I've heard this before. It's not worth asking a question that you're not excited about or interested in or not important because you'll spend a lot of time on that too. So you might as well really ask an important question and tackle it. And I appreciate that that's what they've done here.
So I saw this paper and I said, this paper can be used by the screenwriters of The Last of Us to design the next evil thing.
Why
evil?
Why evil, Michael?
Well, The Last of Us, the Last of Us, these fungi were eating the entire human race and making us all into zombies. So, I mean, it's got a lot of really clever, and you can look at the experiments that you could do to answer these questions to modify the behavior of the worm or the root or whatever you happen to be looking at. And as Mark was going through it, and I know how enthusiastic Mark is about light and light generating microbes, I thought this would be a really cool paper to be part of.
So, Mark, other organisms do bioluminescence. I'm thinking of jellyfish, right?
Yeah.
Mm-hmm.
When you go out in the dock and you push a net in the water, they glow, right?
Yeah. Some of them do. Also, dinoflagellates are mostly due to the, I hate calling it sea sparkle, but most of the really cool stuff where the waves come and break are dinoflagellates. One thing that I always tell people is that bacteria don't blink. And it's true. They produce light consistently or they don't produce it. Right now, they can ramp it up. You know what I mean? But they don't turn it on and off. And where people think that's the case, like the Japanese pinecone fish, Monocentris japonicum, there's a name from the past, that has a Vibrio species, it has a specialized light organ with a shutter that the fish controls. I have never heard of a bacterium that flashes. That would be interesting. But the dinoflagellates do when you disturb them. So...
Oh, that's why it's in the waves. It's not like the waves are glowing all the time. It's only when they're...
Yeah.
Got it.
And when they crash.
And what's the function of that, Mark?
Dinoflagellates doing it? I have no idea with that one.
Or the jellies, right?
Yeah. Now, now I want to be clear. Do you mean bioluminescence or fluorescence?
All right. So it's different. Yeah. It's light, right?
Yeah.
GFP
so one can imagine two different ways of making light, but
right.
They also have different functions depending where, right?
Do you know what the anglerfish is?
Yep.
What I love about anglerfish is it's got its own fish-generated bioluminescence and also bacterial symbiotic ones, both.
Cool.
Well, it's desperate because it's living at depth.
Don't get me started on the solution to that, Michael, because you know what it is, right?
Dilution?
No, Vincent, do you know about this? The males among the anglerfish?
No.
They bite the female, and then they fuse with the female, and then they degenerate until they're only gonads.
Oh, yes. I know that, right.
And you'll find these female anglerfish will have 7 or 8 of these former gentleman collars attached to their corpus.
Got it. Yeah, it's just lovely thought. Okay.
Happy Halloween.
Thank you, Mark.
No, thank you for this opportunity.
Well,
Vincent,
whenever you send a cool paper, you're going to get invited. Okay, so if you want to come back, just send cool papers. Not hard to find. Okay, the show notes for these episodes are at microbe.tv/TWiM. If you enjoy these discussions, how could you not? Please support us, microbe.tv/contribute. And should you have any questions or comments, you want to make a suggestion for something we should talk about, TWiM at microbe.tv. Mark Martin is at Matters Microbial. It's one of our podcasts, and the University of Puget Sound. Thank you, Mark.
You're very welcome.
Michelle Swanson's at the University of Michigan. Thanks, Michelle.
Thank you. And please go vote.
Go vote.
Although it might be too late.
It's too late.
Post.
Right.
Yep.
When this drops, it's too late, probably.
The thought is there. Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levins at Washington University. Thanks, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. See you next time on This Week in Microbiology.